In this video, we will be taking a look at the inhibition of regulatory T cells as a possible method of enhancing anti-tumor immunity. We will focus on the OX40 pathway as a possible target in achieving this outcome. Regulatory T cells, or Treg cells, are important cells in the immune system that serve to keep overactive cytotoxic and helper T cells in check. As a very basic primer in the case that you're not familiar with cytotoxic or helper T cells, cytotoxic T cells directly attack infected or damaged cells, and helper T cells assist in signaling both cytotoxic T cells and B cells. This is a grand oversimplification, but for the purpose of this video, know that they are both essential in clearing infected cells and foreign antigen from the body. In this video, we're going to collectively refer to them as T cells, differentiating them from the cells we're focusing on, Treg cells. Treg cells have the abilities to suppress the development and activity of T cells through a number of different pathways. Normally, this assists the immune system in not over-responding to molecules that T cells encounter on a daily basis, especially molecules native to your body. While we won't delve into the finer details of the typical function of Treg cells, it should be understood that they are essential for the prevention of autoimmune and inflammatory disorders. This brings us to the topic at hand. Normal immune responses are often not sufficient to eliminate tumors from the body, especially due to the fact that tumors establish immunosuppressive environments around themselves. As such, cancer treatment often has to turn to invasive methods like radiation and chemotherapy. However, if the inhibitory action of Treg cells is suppressed, T cells are able to launch a stronger response against tumors, a response that may be able to eradicate a tumor using only the immune system. A target of recent interest in inhibiting Treg cells has been the OX40 receptor. OX40 actually appears on both helper and cytotoxic T cells once they are activated and are essential for promoting their proliferation. On the other hand, OX40 is constitutively expressed on Treg cells and inhibits their suppressive activity when activated. While the mechanism by which this is achieved is still being debated, it has been shown that the activation of OX40 on Treg cells inhibits their production of interleukin 10, a protein that is essential in limiting cytotoxic T cell activity. It has also been suggested that OX40 activation may prevent proliferation of Treg cells in addition to dampening their suppressive activity. This mechanism explains why OX40 targeting may be a useful method of establishing anti-tumor immunity. While T regulatory cells are normally highly active in the immunosuppressive environment around a tumor, activating the OX40 pathway may be able to turn them off and reestablish cytotoxic T cell activity. Now on to how treatment would actually work. Normally, OX40 binds to OX40L, expressed on the surface of antigen-presenting cells, like dendritic cells and B cells. Therapeutic research, however, has focused on instead using a monoclonal antibody to mimic the OX40 ligand. Using monoclonal antibodies, or in other words, an identical group of synthesized antibodies, ensures high accuracy and efficiency in OX40 binding. After treatment with this antibody, activity of induced Treg cells has been shown to decrease. This, along with the stimulatory effect caused by bonding to OX40 on cytotoxic T cells, leads to significantly increased T cell response to tumors. Studies show that in a high number of cases, this can even lead to full tumor regression after cytotoxic T cell attack, with low levels of tumor reoccurrence. Positive results have been demonstrated in both leukemia and breast cancer models. Let's take a look at some experimental results on activation of the OX40 receptor on Treg cells. This study looked at mice infected with cancerous cells and then treated to OX40 monoclonal antibodies. As you can see, the graph depicts tumor size in infected mice, and helper and cytotoxic T cells specifically designed to fight the tumor were injected at day 15. Antibodies were injected on day 15, 20, 24, and 37. The controls were mice not injected with helper or cytotoxic T cells, as shown in diamonds, and mice injected with an irrelevant antibody, shown in square. As seen, treatment with OX40 antibodies, shown in triangles, had hardly any tumor recurrence even at day 100. Without OX40 antibody treatment, Treg cells quickly became effective in suppressing T cells and tumor recurrence was rapidly observed. This proved that lasting anti-tumor immunity was imparted by OX40 antibody treatment. Use of this new therapeutic method has been studied in conjunction with other T cell receptor targets. For example, multiple studies have presented the possibility of activating the OX40 receptor while simultaneously blockading the PD-1 receptor, a T cell receptor that essentially serves as a direct off switch for T cell activity. As shown in this promising experiment, while OX40 antibody treatment and PD-1 blockade alone had little effect on mice infected with ovarian cancer when used separately, combining the two treatments nearly tripled their survival time. Targeting the OX40 receptor to lower regulatory T cell activity, along with blockading other regulatory T cell pathways, serves as a promising look at what the future of cancer immunotherapy may look like.